Are our own cells turning against us? It's strange, isn't it? But it's what we find in our project. So, I'm Adél Lüvi, a fifth-year student in the University Program of National Academy of Scientist Education, and today I would like to present you my work of the role of astrocytes in the development of brain metastasis. So first, raise your hands, please. Who knows what this pink ribbon symbolizes? Okay, there are a few of them. Could someone say it out loudly, please? Breast cancer, thank you. So, pink ribbon symbolizes breast cancer awareness, and the importance of screening exams. The last month, October, was the Breast Cancer Awareness Month. You may have seen some programs dedicated to highlight the importance of breast cancer and to find and detect breast cancer in time. But, why is it so important? Because breast cancer is the most frequent tumor type in women. There are about 2 million newly diagnosed cases each year. It's really important to recognize breast cancer in its early stages because then the primary tumor is very well curable. But an advanced cancer can easily form brain metastasis. There are really aggressive type of breast cancer. It's called the triple-negative breast cancer. It don't express 3 important hormone receptors: estrogen receptor, progesterone receptor, and HER2 receptor. And with this, the treatment of the primary tumor is really hard. This type of tumor is prone to form brain metastasis in about 1/3 of cases. When the tumor cells could once enter the brain, due to the blood-brain barrier, we have really limited treatment options because the blood-brain barrier protects the metastatic tumor cells. It is the first aspect in this presentation where our own cells, our own system turning against us. So let's see how does it work. First, let me introduce you the blood-brain barrier. Under physiological conditions, the role of the blood-brain barrier is to protect the brain from harmful substances. How does it look like? So, in the capillaries of the brain, there are the endothelial cells, and between the endothelial cells, there are so-called tight junctions. These protein-protein interactions, these look like a zipper and work like a zipper, so they connect really hard the cells and make it impossible for most of the substances to enter the brain, even the harmful substances and the metastatic drugs as well. Another interesting type of the blood-brain barrier are the astrocytes. I advise you to remember this name in the last part of the presentation. So, astrocytes cover the vessels with their end feet, as you can see here. So, they are the first who sense the presence of the intruders to the tumor cells. So again, due to the blood-brain barrier, we have limited treatment options in all of the central nervous system diseases. So in the case of breast cancer, brain metastasis as well. In the case of the previously mentioned most aggressive breast cancer type, the triple-negative breast cancer, the prognosis is really shocking. So after the diagnosis, the patient has less than 6 months survival time. It's a few, so it's really shocking. We have to do something against it. As a wise ancient general said, if we get to know our enemies, we will know their weaknesses as well. Following this idea, our research group aimed to uncover the mechanisms behind the formation of breast cancer brain metastasis to find potential points of attack, which may lead one day to treat this devastating disease as well. So, follow me in this presentation. Let me show you our latest results related to breast cancer brain metastasis. There's a clinical study in which the researchers find that there is increased level of the pro-inflammatory cytokine interleukin-1 beta in the blood serum of patients who has breast cancer brain metastasis. We know that interleukin-1 beta is often produced in an inflammasome-dependent manner. But what are the inflammasomes? So the best described part of the inflammasome family is the NLRP3 inflammasome. In the next part of the presentation, I will speaking about this particular inflammasome, but I will refer it just simple as inflammasome. So inflammasomes are part of the innate immune system. They assembled from three main parts: the NLRP3 sensor protein, the AST adaptor protein, and the caspase-1 effector protein. Inflammasomes are activated after pathogen or damage-associated stimuli in two steps. The first step, which is called the priming of the inflammasomes, these pathogen or damage-associated signal molecules binds to pattern recognition receptors, and a signal transduction pathway, so-called NF-kappa B pathway, activates. As a result, in the nuclei, inflammasome-dependent genes are transcribed. After a translation, inflammasome components, so these proteins are produced: the NLRP3 sensor protein, the adaptor protein, the pro-caspase-1 effector protein, and also the pro-form of the cytokine, the pro-interleukin-1 beta. The next step, which is the activation of inflammasomes, these inflammasome components assembled into an active inflammasome. During this process, caspase-1 is active, and it cleaves pro-interleukin-1 beta to active interleukin-1 beta, which is one of the most potent pro-inflammatory cytokines in the human body. During our project, we wanted to answer three main questions. So first, we were curious about whether inflammasomes play any role in the formation of brain metastasis. If the answer is yes, we wanted to know which cell type is responsible for inflammasome production. And finally, we wanted to know whether inflammasomes have any effect on metastatic tumor cells. To answer these questions, first, we had to create a brain metastasis model. For this, we injected mouse breast cancer cells into the right carotid artery of biopsy mice. Here, see. With this, we achieved that the tumor cells were growing only in the right hemisphere of mice, so we could use the left one as control in our experiments. To detect and visualize the proteins of interest, we used immunofluorescence staining. For this, we fixed the mice brains, we made coronary sections, and labeled them with a specific antibody, and we used confocal microscopy to detect the signals. We also used another technique. It's called Western blot technique, which is used for the semi-quantitative measurement of proteins. We use this to measure the level of the active cytokine, the active interleukin-1 beta. So first, in order to detect the localization of the inflammasome components in mice brain metastasis, we labeled the NLRP3 sensor protein and the interleukin-1 beta in green, and the astrocytes in white. You can see in these pictures quite well that both the NLRP3 signal and the interleukin-1 beta signal co-localizes with the astrocyte signal. But there are no signals in the tumor cells or any other stromal cells. So, sorry, I forgot to mention that you can see the tumor cells pink or in red in these pictures. So we can say that the inflammasome components are specifically in peritumoral astrocytes. If you remember, I mentioned you that the astrocytes are the first who sense the presence of tumor cells. So we can say that this is their first reaction to the metastasis formation. We also observed that the interleukin-1 beta expression level correlated with the size of the tumor cells. So here you can see the larger the tumor lesion is, the more interleukin-1 beta are produced. Then we wanted to follow the inflammatory process in breast cancer brain metastasis formation. So we performed immunofluorescence staining 2, 5, and 7th day after the injection of tumor cells. So 2 days after the injection of tumor cells, there is no visible interleukin-1 beta staining in the brain, because at this point, the tumor cells are still stuck in the lumen of brain vessels. They need time to open the really tight zipper-like blood-brain barrier. But after 5 days, when the tumor cells have already extravasated and entered the brain, the interleukin-1 beta signal appeared, which increased in the 7th day. And you can see that it still colocalizes with our astrocyte signal. Then we wanted to uncover the role of NLRP3 inflammasomes. So as far we know from the immunofluorescence staining that inflammasome components are upregulated in peritumoral astrocytes. But we wanted to know the role, so we inhibited the inflammasomes with a specific NLRP3 inflammasome inhibitor called MCC950. So after the injection of tumor cells, in the 4th, 5th, and 6th days, we treated the mice intraperitoneally, so practically we injected these drugs into their belly. And on the 7th day, we perfused them. And we used their brain for further experiments. For example, for protein measurements. For this, we separated their brain because, as I mentioned before, the right hemisphere is the one which contains the tumor lesions. We homogenated the brain samples, and here we created 4 samples. So the control mice control side and tumor-injected hemisphere, and the MCC950 treated mice control side and tumor-injected hemisphere. In the following part of the presentation, you will see this 4 groups in the same order, colored with the same colors. So control with green and MCC950 treated with gold. So in this picture, you can see a representative Western blot in which is very well visible that there are increased level of active, so the cleaved, the shorter form of interleukin-1 beta in the control mice tumor-injected hemisphere. We used densitometric analysis to this band. This is how we made this chart. And it's really well visible that, yes, again, there are increased level of interleukin-1 beta, active interleukin-1 beta in the control mice tumor-injected hemisphere, but MCC950 could significantly decrease it in the MCC950 treated mice tumor-injected hemisphere. So, as a final part, we just wanted to be sure that the astrocytes are the cell types who, so in which the inflammasome components are activated. So for this, we used immunofluorescence staining. for interleukin-1 beta in the same setup as I mentioned before. So we treated mice, so there are control mice and MCC950 treated mice. Both of them had control and tumor-injected hemisphere. So it's really well, it's really well visible here as well that yes, there is increased level of interleukin-1 beta in the control mice tumor-injected hemisphere. But there is a visible decreasing of interleukin-1 beta signal in the mice which were treated with MCC950. And most importantly, we observed that not just the level of interleukin-1 beta, but the size and but the size of the tumor cells, the tumor lesions, were also decreased. So when we quantify these pictures, we came to the result that the tumor cover pixels were decreased more than a half in the MCC950 treated mice, which means that there were significant smaller and fewer tumors in the mice which were treated with MCC950 inflammasome inhibitor. To shortly sum up our results, we found that the expression of inflammasome components is restricted in peritumoral astrocytes. These inflammasomes produce interleukin-1 beta, which increases the proliferation of tumor cells in the brain. And this is the final aspect or final, this is the second aspect or final conclusion when our own system or our cells turning against us, and they produce inflammatory factors which helps the proliferation of breast cancer brain metastases. But with the inhibition of inflammasomes, we could decrease the number and the size of tumors in the brain. So as a final conclusion, we can say that the NLRP3 inflammasome could be an effective therapeutic target in breast cancer brain metastases. I'm really grateful for my mentor, Dr. Imola Wilhelm, my colleagues, Dr. Kinga Mészáros-Molnár, Dr. Csilla Fazekas, Dr. Ádám Mészáros, our group leader, Dr. István Kriszbai, and every member of the Neurovascular Unit Research Group for their lots of support and help. I thank for the organizers for the opportunity to present here today. And I also thank you for the kind attention. And if you have any questions, please feel free to ask in the upcoming coffee break. Thank you.